So here I have this little diagram to explain the Nirenberg and Matthaei experiment that was done in 1961. The premise of this experiment was to use homopolymers for U, C, G, and A to try and figure out the corresponding amino acid to those codon sequences. And so they started off with this test tube right here, and it had three different components. It had the cell-free translation system, which is basically everything in the cell that could still synthesize proteins and go on business as usual without having the cell wall around it. They also had the radioactive amino acid that was labeled, which was different in each of the many trials that they made. And they also had a poly-U homopolymer, which is a string of U's that was used for this specific trial here. After that, it was incubated at about 37 degrees Celsius, if I got that right, and then translation would occur. And if the amino acid that they had labeled was incorporated into this translation, it would synthesize a protein, which you'll see here in blue dots. If not, the solution would just stay clear with no protein there. Afterwards, they did section and other things that I'm not going to go into. And I assume with a lot of patients, they went through 20 different trials with a different amino acid labeled in each trial. For example, this would be proline, serine, etc. The only result they got was in phenylalanine for the poly-U homopolymer. Some results from this specific experiment was that they determined 3 out of the 64 codon combinations that they had known. They got phenylalanine for the poly-U, proline for the poly-C, lysine for the poly-A, and could not get the poly-G to work, but they would have gotten glycine. The second experiment that was done by Nirenberg and Leder is where they used short kind of mRNA strands with different codons or trinucleotides on them, and they put them in a mixture of ribosomes and radioactively labeled amino acids. And then the goal of that was to kind of have the mRNA match up with the tRNA. And if that radioactive amino acid was used when coding for this trinucleotide, it would fall onto the filter paper that they had at the end of the experiment. And this is actually a little diagram of translation that I did for the Nirenberg and Matthaei, not